Plants nutrients. A. General classification. 1. Major or macronutrients. Those nutrients which are required by the plants in concentrations more than 100 ppm are termed as major or macronutrients. These are grouped into A. Basic nutrients. C. H. O provides basic structure to plants. B. Primary nutrient. N. P. K. C. Secondary. C. A. M. G. S. 2. Minor or micronutrients. The elements which are required by plants in concentration less than 100 ppm are put in this category. They are also called as trace elements or oligo elements. These are Fe, Mn, zinc, Cu, Bo, Cl and molybdenum. 3. Amobile nutrients, Ca and B. 4. Energy exchange, H and O. 5. Cation nutrients, K, Ca, Mg, Fe, Mn, Cu, zinc. 6. Anion nutrients, P, B, Mo, Cl, So 4. 7. Both anion and cation form, NH4+, NO3. 8. Non-metals nutrients, B. B. Other classification of plant nutrients. 1. Essential nutrient. According to Anand and Stout, there are 16 essential nutrients for plants, e.g. C, H, O, N, P, K, Ca, Mg, S, Fe, Mn, Mo, B, zinc, Cu, Cl. Nickel is the 17th essential element known in 1987 by Brown et al. Criteria of essentiality of plant nutrients. Given by Anand and Stout in 1939. They stated that plant can't be completed their life cycle without the deficiency or complete absence of a single nutrient among the essential nutrients. Anand, 1954, refined the criteria of essentiality of nutrients. Leaving law of minimum. According to Justus von Liebig, 1840. The growth of plants is limited by the plant nutrient present in smaller quantity, all others being in adequate amounts. This has been restated as barrel concept. 2. Beneficial nutrients are 4 in number. Vanadium, V, silicon, C, cobalt, Co, sodium, Na. 3. Functional nutrients. The term functional nutrient was proposed by D. J. Nicholas 1963. Functional nutrients are 20 equals essential nutrients 16 plus beneficial nutrients CO, V, C, Na 4. Includes all 16 essential nutrients in addition to 4 beneficial nutrients CO, V, C, Na, collectively known as functional nutrients. Essential elements and their form of availability. SN, element concentrate in plants, form of uptake. 1. Major nutrients. Carbon, C, 45% CO2. Hydrogen, H, 6% H2O. Oxygen, 45% H2O, O2. A. Primary nutrients. Nitrogen, N, 1.5% NO-3, mainly NH plus 4. Phosphorus, P, 0.2% H2PO4, mainly HPO2 to 4. Potassium, K, 1.0% K plus. B. Secondary nutrients. Calcium, Ca, 0.5% Ca2 plus. Magnesium, Mg, 0.2% Mg2 plus. Sulfur, S, 0.1% SO2-4, SO2. 2. Micronutrients. Iron, Fe, 100 ppm, 0.01%, Fe2 plus, mainly, Fe3 plus. Zinc, 20 ppm, Zinc 2 plus. Manganese, Mn, 50 ppm, Mn2 plus. Boron, B, 20 ppm, H3BO3, boric acid. Copper, Cu, 6 ppm, Cu2 plus. Molybdenum, Mo, less than 1 ppm, 0.1 ppm, Mo042. Chlorine, Cl, 100 ppm, Cl. Nickel, Ni, 0.1-1.0 ppm, Ni2 plus. In acidic soils, phosphorus is available in H2PO4 form, and at alkaline, high pH, phosphorus is available in PO4-3 form. Most of plants absorb nutrients in reduced form, e.g. Fe+2 and Mn+2, rather than oxidized form, e.g. Fe+3 and Mn+3. Soil test rating chart for available nutrients. Nutrients, compounds, units, rating, low, medium, high. Carbon, organic carbon, percent, less than 0.5 to 0.5 minus 0.75 greater than 0.75. Nitrogen, alkaline KMNO4, kilogram per hectare, less than 250, 500 greater than 500. Phosphorus, Olsen's method, P kilogram per hectare, less than 12.5 to 12.5, 25 greater than 25. Phosphorus, Olsen's method, P2O5 kg per hectare, less than 28, 56 to 56. Potassium, ammonium acetate, kilogram per hectare, less than 140 to 140 minus 280 to 280. Calcium, ammonium acetate, CMO, P+, kilogram, less than 1.5 to 1.5. Sulfur 0.01 mccal, kilogram per hectare, less than 22.4 to 22.4, 35 greater than 35. Magnesium, ammonium acetate, CMO, P+, kilogram, less than 1.0 greater than 1.0. Zinc, DTPA extractable, milligram per kilogram, less than 0.6 to 0.6 minus 1.2 greater than 1.2. Iron, Fe, DTPA extractable, milligram per kilogram, less than 5 to 10, greater than 10. Manganese, Mn, DTPA extractable, milligram per kilogram, less than 5, minus 5, 10, greater than 10. Copper DTPA extractable, milligram per kilogram, less than 0.2, 0.2 minus 0.4, to 0.4. Boron, hot water soluble, kilogram per hectare, less than 0.5, minus 0.5 minus 1.0, to 1.0. Molybdenum, ammonium oxalate extractable, pH 3.3, milligram per kilogram, less than 0.2, minus 0.2 minus 0.4, to 0.4. Role of nutrient. 1. Nitrogen. Impart green color to plant, encourages vegetative growth. It is present in most of substances of cells. Nitrogen is essential constituent of protein. Constituent of protoplasm of chlorophyll and coenzyme. Play important role in synthesis of auxin. Deficiency. Lower leaves become yellow or light green in color. V-shaped chlorosis on older leaves or yellowing at tip. Disease. Starvation disease due to nitrogen deficiency. Buttoning in cauliflower. 2. Phosphorus. Increase the disease resistance. Enhance new cell formation and necessary for root development. Required for grain formation and maturity of grains. Phosphorus is essential constituent for nucleic acid and phytin. Most essential functions are energy storage and transfer of energy, ADP and ATP, act as energy currency. Deficiency. Due to deficiency of single element, phosphorus, the life cycle of plant can't be completed, hence, phosphorus is called as key of life. Deficiency imparts dark green color in leaves. Bronzing appearance on older leaves. Later develops red-purple coloration on leaves of maize. Disease. Sickle leaf disease. 3. Potassium. Most essential function of K is stomata regulation. Provides disease and drought tolerance in plants. Co-factors for enzymes. Responsible for quality products. Formation and translocation of sugars. Helps in chlorophyll formation. In plants, K+ also reduce the transpiration rate and increase photosynthetic rate. Because it is stress avoider. Potash useful in stress condition because it secretes 60 enzymes. The term luxury consumption is often used to describe nutrient absorption by the plant that does not influence yield, e.g. potassium. Deficiency symptoms. Spot of dead tissue at tips of leaves. Scorching and burning on margins of bottom leaves. Disease. Show rosette or dieback discase. Calcium. It is constituent of cell wall. Calcium are mobile in plants and deficient symptoms appear on meristem tip portion. Deficiency. Terminal bud may finally die. Disease. Tip hooking and curled margins of leaves. Blossom end rot, bare, disease of tomato. Magnesium, Mg. Essential constituent of chlorophyll. Magnesium is a central atom of chlorophyll. Leaves become mottled and chlorotic, then reddish or upward cupping of leaf. Disease. Sand drown, disease of tobacco. Sulfur. It is essential constituent in some amino acid, i.e. cysteine, cysteine and methionine. Sulfur oxidizing bacteria is Theobacillus. Sulfur is essential for oil seeds and pulses because it improves oil content and protein content in oil seeds and pulses, respectively. Thiourea at 1 gram per liter spray on standing crops, best for N and S deficiency. Deficiency. Deficiency symptoms resemble to those of N deficiency, but young leaves are pale green or light yellow without any spot. Disease. Akiochi of rice due to excess of H2S. Tea yellow, disease of tea. Iron, Fe. Required for nitrogen fixation because it catalyst of chlorophyll synthesis. During respiration act as O carrier, leg hemoglobin. Act as enzyme in oxidation reduction process. Deficiency. Interveinal complete chlorosis but main vein remain characteristic green. Scorching of leaf margin. Young leaves become yellowing or iron chlorosis in groundnut. Manganese, Mn. Formation of chlorophyll. Cofactor of enzyme. Mn toxicity causes crinkled leaf of cotton. Deficiency. Dead spot on leaves. Leaves become chlorotic but smaller vein remain green. Diseases. Pahala blight of sugarcane. Gray spike of barley, oat. Mass spot of pea. Copper, Cu. Tips and margin of young leaves show chlorosis. It is compound of plastocyanin. Essential for photosynthesis, respiration. Rosetting and excess gumming occurs due to copper deficiency. Gamosis and xanthomania disease of citrus. Dieback and reclamation disease of citrus due to deficiency of copper. Boron, B. It is necessary for pollen germination. Boron is the only non-metal element among the micronutrient. It is necessary for translocation of sugars and is involved in reproduction and germination of pollens. It concerned with water reactions in cells and regulates the intake of water into cell. It may act as regulator of potassium ratio and keep calcium in soluble form within the plants. Taste in cauliflower due to presence of boron. Diseases due to deficiency. Browning or hollow stem of cauliflower. Top sickness of tobacco. Dieback and rosette of apples. Fruit cracking of tomato and pomegranate. Hard fruit of citrus. Hen and chicken disease of grapes. Heart rot of sugar beet. Zinc. In plants it's required for biosynthesis of hormones. Deficiency causes. White bud of maize. Khariya discase of rice. Little leaf of cotton and apple. Mottled leaf of citrus. Rosette formation. Cobalt, Co. It is component of vitamin B12. It is essential for formation of a type of hemoglobin in N-fixing nodule tissue known as leg hemoglobin. It is act as O2 carrier in roots. Also known as animal protein factor. Molybdenum, Mo. It absorbed as molybdate, Mo O4-2 forms. Mo is essential component of enzyme, nitrate reductase. Functions as N-fixation in pulses. Whiptail of cauliflower is due to deficiency of Mo. Mo is required for carrot and raffinus for sweetness. Chlorine. Absorbed as Cl form. High Cl sensitivity crop, legume and tobacco. Other facts. Fruit cracking of tomato is due to deficiency of boron. Reclamation disease, also called white tip, is caused due to deficiency of copper. Sodium, Na, is important constituent of cam plants. Silicon absorbed by plants as monosilicic acid and it is essential for rice. Pollen grain sterility of wheat is due to deficiency of boron. Sodium is essential for osmoregulation in plants. Sodium is responsible element for drought tolerance in sugar beet. Chlorosis between the veins and veins remain green shows deficiency of Mg. Complete intervenal chlorosis occurs due to Mn deficiency. In iron deficiency, the principal veins remain conspicuously green and other portions of the leaf turn yellow, tending towards whiteness. In manganese deficiency, the principal veins as well as the smaller veins are green. Silicon is essential for rice and maize. Black heart of potato is due to deficiency of oxygen. Metabolic transformation of inorganic plant nutrients into organic plant constituents is known as assimilation.